Boa tarde a todos e a todas. É um prazer grande estar aqui nessa reunião da SOBEP, no canal do YouTube, com a doutora Caroline Shiboski, que vai apresentar a palestra intitulada Sjgrens International Collaborative Clinical Alliance, Lessons Learned in Future Directions. É com grande prazer que temos conosco a doutora Caroline nessas atividades que estão se tornando rotina na nossa sociedade, nos encontros mensais aos sábados. Hoje, particularmente, devido à questão de fuso horário, estamos começando num horário um pouco diferente, mas com certeza será uma atividade enriquecedora para todos nós. A professora Caroline é professora de Medicina Oral e chefe do Departamento de Ciências Orofaciais da Faculdade de Odontologia da Universidade da Califórnia, em São Francisco. Ela é graduada em odontologia na França, na Université René Descartes, em Paris, com treinamento em medicina oral na UCSF, sendo diplomada pelo American Board of Oral Medicine, e treinamento em epidemiologia, com mestrado e PhD pela University of California, Berkeley. Suas atividades de pesquisa têm por foco a epidemiologia de manifestações orais relacionadas a disfunções imunes, ao câncer bucal e, particularmente, a síndrome de Sjögren. A doutora Shiboski tem recebido financiamento de forma constante do Instituto Nacional, do National Institutes of Health, o NIH, desde 1994, como na forma de pesquisadora principal, e hoje ela vai abordar as lições aprendidas pela Sjgrens International Collaborative Clinical Alliance, a SICA, o maior biorepositório e registro de dados sobre a doença Sjögren no mundo, e também comentar sobre os novos projetos que se originam dessa colaboração. Caroline, it's a great pleasure to have you here with us. And please, it's your talk.
Thank you very much, Luis. I really appreciate the invitation, and I'm delighted to be here with your colleagues this morning and tell you more about SICA, the Sjogren's International Collaborative Clinical Alliance, which actually started in 2003. So it's been about 19 years. So as you all know, Sjogren syndrome, which is actually now called Sjogren disease, and there's going to be a whole international vote on this on this change, but it was started by a letter from Alan Bayer and Kathy Hammett in arthritis and rheumatology. Sjogren disease is a multi-system autoimmune disease that's mainly characterized by lymphocytic infiltration of exocrine glands. And so you get significant loss of secretory function, and it affects predominantly salivary and lacrimal glands, which is why we're so involved as oral medicine clinicians. It is the most common autoimmune disorder in the US after rheumatoid arthritis, and it affects predominantly women in a 9-to-1 ratio. And it has also a number of systemic features that include inflammatory arthritis, immune thrombocytopenia, vasculitis with purpura, salivary gland enlargement, a number of neuropathies, as well as interstitial lung disease and primary biliary cirrhosis. So the risk of lymphoma with Sjogren disease is up to 19 times higher in patients with the disease compared to the general population. And also early and aggressive immunomodulation systemic therapy that's recommended for rheumatoid arthritis and other autoimmune disease has not been well established for patients with Sjogren disease. So essentially there is no effective treatment. And to date, the management of Sjogren, which by the way requires multidisciplinary approach given its heterogeneity. So you need rheumatologist, ophthalmologist, and oral medicine clinician. The management is predominantly palliative and symptom driven. So for oral medicine clinician, you all recognize the very dry mouth, very dry tongue, sometimes this discrete patchy erythema, which can reflect erythematous candidiasis. Salivary gland enlargement, which is present in maybe about 15 to 20% of patients with Sjogren, as well as rampant caries that affect mainly smooth surfaces. So we manage, at our end of things, we manage these patients obviously with topical fluoride to make sure that they do not develop rampant caries, and then trying to control the recurrent oral candidiasis, which is often an issue in patients with such dry mouth. So let me tell you a little bit about how SICA, or the Sjogren's International Collaborative Clinical Alliance, came to be about. So it was founded in 2003 through a contract by NIDCR, and the initial co-directors or co-PIs were Troy Daniels and John Greenspan, and I'm sure many of you have heard these names. I was brought into the team as a lead epidemiologist, and my colleague Lindsay Criswell was brought in, who is a rheumatologist, was brought in because of her expertise in genetics. And Stephen Shiboski, who happens to be my husband, was brought in to lead the data center that was to become part of this whole project. So the goals of the project were to develop new classification criteria for Sjogren disease, to better characterize the Sjogren disease phenotype and genotype, and to establish a data biorepository to support pathogenesis, epidemiologic, and genetic studies on Sjogren disease, and have a dissemination plan for worldwide access. So any investigator in the world who has a project as part of which they would like to acquire biospecimens related to Sjogren could actually come to us to obtain these specimens. And we started this international project with a number of colleagues around the world, and this was greatly due to both Troy Daniels and John Greenspan's connections. Troy had worked in Sjogren's for like 40 years and knew many people around the world working on this disease. So we had a group in Argentina that was led by Hector Lanfranchi. We had a group in China. We had a group in Denmark led by Morten Schut. We had a group in Japan. Our main headquarters were in San Francisco. A little later on, we had a group that joined us from King's College London in the UK that was led by Steve Shalacombe. In Yerevan, India, these were the latest to join us, and then two other groups in the US, one at UPenn and one at Johns Hopkins. Actually, I wonder if I can do the full slide. Yes, this is better. So this is our group in 2009 as we would get together on a regular basis to obviously at the beginning to decide which items we were going to include. I mean, what we were going to measure on our participants, who we would recruit. And so you might recognize here in the front row, John Greenspan, Troy Daniels. This is Lindsay Criswell, and this is myself. And then these are all our other collaborators. So for the enrollment criteria, we used a fairly large net because we wanted adults, patients 21 years or older, who had a complaint or a sign that could suggest that they might have or develop Sjogren disease. So they may have a complaint of dry eyes or dry mouth. They may have had a previous diagnosis of Sjogren, or maybe they had salivary gland enlargement, et cetera, et cetera. And these were the tests we all agreed we would be collecting on these participants. So for the eye, we collected a Schirmer test, a tear breakup time. Then the ophthalmology group, one thing that I forgot to mention when I introduced the group is that each of the 9 research groups had a rheumatologist, an ophthalmologist, and an oral medicine clinician or an oral pathologist included in that group. So it was truly multidisciplinary, and the multidisciplinary expertise was available in every single research group. So all the ophthalmologists from all 9 groups together developed what came to be known as the OSS, the ocular surface stain, that was done with lissamine green and fluorescein, and I'll talk a little bit more about it in a minute. Then from the oral salivary standpoint, we collected both unstimulated whole saliva as well as stimulated parotid flow rate with a little suction cups. And we also collected labial salivary gland biopsy, both so we sent about 4 or 5 glands for H&E, so that we could actually get the diagnosis read by our histopathologist. We were looking for focal lymphocytic cellulitis with focus score, and then the rest of the glands, and we tried to collect on average 8 to 10 glands per participant. The rest of the glands were flash frozen and then stored in our biorepository. Rheumatologically, we did a physical exam that was done by a rheumatologist and collected a number of serologies, as you can see down here. And then the biospecimen stored included serum, plasma. We had collected whole blood from which we extracted DNA. So we have DNA on all our participants. We had whole and parotid saliva that was also stored, tears, as I mentioned before, the labial salivary glands, both frozen and paraffin embedded, conjunctival RNA, and then PBMCs on the subset of participants who had enrolled at UCSF, because it's quite expensive and actually delicate to try to process PBMCs, and it can be difficult to ship from one site to the other. So we decided to do it only on one site. And I apologize, somehow the formatting of this slides sort of got lost, I think, in the transfer. So basically this should show you that the SICA cohort enrolled a total of 3,514 participants across all sites. You can just see the percent here. So 37% came from the US, one of the 3 sites in the US, 17% from Denmark, 13% from Argentina, and etc. And these are all the referral sources. So patients were recruited either from Sjögren syndrome clinics, from rheumatologists, from dentists, ophthalmologists, etc. And then we did have a follow-up visit after 2 to 3 years. So anyone who had at least one positive objective sign was invited to come back, and about a little shy of 800 presented for a follow-up. And everyone who came for a follow-up had a repeat of every single test and questionnaire that had been asked previously. So I previously showed you the type of clinical data that was collected. In addition to the clinical data that came to basically be in our biorepository, we also had a number of questionnaires, oral history, medical history, and a number of other questionnaires which were translated into the local language of each of the research site and back translated to make sure that nothing got lost in translation, and so that all of our data would be as uniform as possible. So with this kind of project, the phenotypic data is extremely important, and you really want the same way of collecting this phenotypic data across the entire project. So among our 3,514 participants, you can see that, not surprisingly, the vast majority were women, 91%. In terms of age distribution, while we had the majority of the participants that were over the age of 50, we still had a fair number of younger participants. And in terms of race, the 2 most common races were white participants and Asian, because of our the involvement of Japan and China and India. So this was one of the first projects that all the ophthalmologists worked on, which was the development of this simplified quantitative method for assessing choroconjunctivitis sicca. And as you can see, not only is the conjunctiva stained with lissamine green, but also the cornea is stained with fluorescein. And the denser, if you will, the stain uptake, the drier the eye is. And this was actually recruited on a form. And you can see that there's a certain number of points that go with how many dots can basically be estimated on both the temporal and nasal conjunctiva, and then a number of points for the cornea. And all of this essentially, and you also get an additional point for patches of confluent staining, if there is staining in the pupillary area, and also for the presence of filaments. So all of this can add up to a score which is at the most 12. And so you would have a score between 0 and 12 for each of the two eyes. So very early on, and as you can see, we only had, this was in prior to 2010, so we didn't have all of our participants recruited yet, but we tried to visualize our data using this area proportional Venn diagram so that we could better understand the interrelationship between the OSS, the labial salivary gland diagnosis of FLS with focus score greater than 1, and then the green circle here allowed us represented the positive anti-SSA or anti-Rho or B or La antibodies, and this was basically sort of preliminary work that was done to try to figure out what the right cutoffs would be and how these different phenotypic features interacted with each other. And one thing that you can see is that whether you use the OSS or the Sicca score represent the ocular staining score, if we put it at a threshold of 3 or at the threshold of 4, we still have a number of participants here that basically only have a positive OSS and none of the other two features. And this is also reflected if you look at the distribution of the OSS on a graph. You can see that on this curve here, which represents the participants with what's called KCS, choroconjunctivitis sicca only, and none of the other features of Sjgren, they tend to have, their peak is sort of closer to 3, whereas the ones who have other features of Sjgren's, which we call SSKCS, the peak here is much higher, and the OSS is supposed to be much higher. Then we also had some preliminary work done led by Troy Daniels, and in which we explored associations between the salivary gland histopathologic diagnoses and the phenotypic features of Sjgren's. And again, all of these analyses were done essentially at some point in time. So we were not completely done recruiting all our participants. So this particular one involved about 1,700 registry participants. And so I don't need to remind this audience that includes a majority of oral pathologists how focal lymphocytic cellulitis basically appears on the salivary gland with these foci of lymphocytes, and how the focus score is computed, and the threshold for that focus score was set at greater or equal to 1, as has been done sort of throughout the years in previous, in the literature. So for example, if you look at the American European consensus group criteria, this was also the cutoff for the focus score. So you can see that the odds ratio here are extremely high. So the odds of having positive anti-SSA or B serology is 9 times higher in somebody who has FLS with a focus score greater or equal to 1 compared with somebody who has the lower focus score less than 1 or chronic silodenitis or some other histopathology in the salivary gland. And this is also true for the OSS. So the ophthalmologists decided to set the OSS at 3 because they wanted to make sure that they did not use too stringent a cutoff. So even though the epidemiologist and the statistician, based on the graph, we could see that the OSS probably should be quite a bit higher based on the interrelationship with other phenotypic features. They ended up setting it at 3 at first, which is the first sort of abnormal score, if you will. So they just wanted to set it as an abnormal score. And you'll see that actually ended up changing over time. And the unstimulated whole saliva flow rate, which actually should be less or equal to 0.1 ml per minute. Again, people with FLS and a higher focus score were two times higher, with twice the probability of having a lower unstimulated whole saliva. And then there was no significant association with the symptoms of dry eyes and dry mouth, which, as you know, can be quite non-specific. Now, another interesting analysis that was done, and I'm not presenting this in a way in a linear fashion. A lot of these analyses sort of happened over time. So for instance, this particular one was done in a way after we initially published our first classification criteria. But I think it's important because it will affect the second set of criteria we ended up coming up with. So this particular analysis, which was led by Alan Baer at Johns Hopkins, and that enrolled almost 3,300 participants, found that 35% had anti-SSA with or without anti-SSB, but only 2% of these participants had anti-SSB or LA alone. And the presence of anti-SSB without anti-SSA had no significant association with Sjogren disease phenotypic features when compared to seronegative participants. And this actually led to the elimination of anti-SSB as a criteria item in the later iteration of our criteria, which I will talk about in a few minutes. Then we also had the opportunity to look at the change in phenotypic feature over time. Now, you might all comment on why 2 years? 2 to 3 years seems like a very short period of time to do a follow-up on patients with a chronic disease, especially a disease like Sjogren's. But this was actually constricted by the funding. So the initial funding was 5 years. And within 5 years, basically, if we wanted to do a follow-up, and the follow-up was actually mainly to try to follow people who did not meet the criteria baseline, was there any change basically within 2 years of people who sort of would actually meet criteria within 2 years? And we did find, so we found a great stability of the majority of items over time. But we did have a certain number of people who basically sort of converted. So among 317 people who were negative at study entry and became positive 2 years follow-up, what we found is that certain serologic markers were predictors of this progression. So, for instance, if somebody had hypergammaglobulinemia or an IgG greater than this threshold here, these people were almost 4 times more likely to basically become positive for Sjogren. And in the same way, people who had hypocomplementemia or a C4 level less than 16, these people were 6 times times more likely to basically become positive. And these were all statistically significant. We had a couple of case reports that came out of this. Actually, the malt lymphoma that was diagnosed in a labial salivary gland from one of the participants we ended up publishing it because it was relatively unusual. Unlike other cohorts, we had relatively few lymphomas. Less than 1% of our participants developed lymphoma. And then we also had another case that was worth publishing in that we had one participant that was diagnosed with IgG4 related disease from the lip biopsy. So this is an example of one of the what we call dissemination plan projects. So a group of investigators from Johns Hopkins, this group was led by Anthony Rosen and Livia Cassiola-Rosen, obtained data from SICA as part of our dissemination plan. And they were interested in the idea that activation of distinct inflammatory pathway may drive the disease heterogeneity. So their study aim was to investigate whether activation of the interferon pathway correlates with key phenotypic features. And so they requested labial salivary glands and clinical data from 82 SICA participants. They had 53 Sjgren and 29 non-Sjgren. And they found that 58% of these participants had high interferon activity and that they differed significantly from those with low interferon activity. They had higher prevalence of abnormal findings on the unstimulated whole saliva. They had leukopenia, hyperglycemia, high titer ANA, anti-SSA, and high focus score on the lip biopsy. There were 3 distinct patterns of interferon: a type 1 predominant, type 2 predominant, and a type 1-2 mixed interferon. And the focus score was highest in those with the type 2 predominant interferon. So the associations between the interferon pathways and disease severity suggests, or disease activity suggests, that interferons are relevant therapeutic targets in Sjogren's syndrome. So this is just to give you an example of the type of project that can be done when you obtain data from the SICA biorepository. This was another example of data obtained. And you can see both my name and Lindsay Criswell's name on both of these papers. It doesn't mean that we always collaborate. I mean, basically, it depends on the people obtaining the data. Sometimes they like to have us collaborate because we obviously have a lot of expertise in the phenotypic feature of Sjogren. So sometimes we're asked to collaborate and look at the analyses, but this is certainly not a requirement, and people can very well obtain our data. There is a process by which we send proposals out for external review so that it's an objective process, and we first make sure that we have the specimens that people request, and then we sent out the proposal for external review, essentially for sort of scientific validity. And once it's been approved by the external reviewer, then we go ahead and share the specimen. So this was an example. This was done by a group at UCSF, and they did mass spectrometry analyses that showed that Sjögren disease is associated with increased N-glycosylation of numerous salivary glycoproteins in both parotid and whole saliva. And the observed alterations in the salivary proteome and the N-glycome could potentially be used as Sjögren disease biomarkers. And this would enable easier and earlier detection of Sjögren. So again, an example of a project which was done with some of our biospecimens. So now, this was basically, I told you about the primary goal of SICA, which was to develop classification criteria. And we came up with an initial set of classification criteria based on a data-driven expert consensus approach. And we published this in April 2012. We were very happy because this was actually approved by the American College of Rheumatology. And so they became the ACR classification criteria for Sjogren syndrome. Now, the approval was actually provisional, as you can see here, which I've highlighted, because they wanted us to validate the criteria in an external data set. And so we still needed to do that. And we approached one of our French colleagues, Xavier Mariette, who is a rheumatologist in Paris and who has a large cohort of Sjogren's patients in Paris, and asked him if he would be willing to have us validate the classification criteria in his cohort. And we were surprised. Xavier actually laughed and said, you have to realize that people are very upset. People in the European scientific community are quite upset with these new criteria because they have been using the American European consensus group criteria, which were developed probably 10 years prior, but had not been endorsed by either ACR or EULAR. But they were very happy with these criteria. And so now we had this new set, and it was annoying to the scientific community to have two sets of criteria for Sjogren's. So we decided to create a working group. Instead of fighting over whose criteria were going to be used, it was much better to work together. So we created the ACR EULAR working group on Sjogren syndrome classification criteria. And our objective was to refine the provisional ACR and the AECG criteria to a single consensus formulation to be endorsed by both ACR and EULAR. So EULAR used to stand for the European League Against Rheumatism. And I remind you that was actually not created until after SICA had started, which is why initially we were mainly seeking ACR approval. But now that EULAR existed, obviously we wanted the criteria to be approved by both, because we wanted these criteria to be accepted worldwide. The criteria are important because they essentially allow you to enroll patients into clinical trials. And they are very important in essentially making sure there's no misclassification when you enroll patients into clinical trial. And it's particularly important when testing drugs, biological agents, like, for example, like rituximab, or these drugs should definitely not be given to people who are not thought to have the disease. And so hence the importance of the classification criteria. And the Sjögren Foundation supported most of our meeting and helped us organize most of the meeting with this working group. So there's a whole formal process. We presented this proposal that was approved by ACR and EULAR in 2014. So we had to have like a steering committee with coordinators, co-conveners, methodologists, and so you could see Steve Shiboski was a statistician, Raphael Seror, who's a French rheumatologist, epidemiologist, and myself were the epidemiologist. And then we had 52 clinician experts from 14 countries and 4 continents, and two patient advocates, both from France and from the US. And we decided to actually use and this was just in an initial step, we used this software that's called 1,000 Minds. And this was developed by 2 mathematicians, actually mathematician econometrists. And the software was developed basically as a method for prioritizing patients for elective surgery. But we quickly, when I say we, it's a royal we, the rheumatology group realized that this could also be used an initial step to quantify consensus when working on classification criteria. So this had been used for the development of classification criteria for systemic sclerosis as well as for RA. So in a way, we kind of had a blueprint, and we decided to try to follow what had been used for other rheumatic diseases. And what the multi-criteria decision analysis does, it's basically based on pairwise ranking of alternatives, and you essentially feed a software a number of items which you think are going to be of interest in the disease of study. And then the software will generate a number of pairwise vignettes. And that survey is basically administered to a large number of experts. And each of them will essentially fill out, choose in these vignettes. And in the end, the software will come up with so it's an iterative process, and the software comes up with a weighted a weight for each of the items. So and I'll show you an example. It's a little easier to look at an example. So this is an example of a vignette, and the expert would basically choose, well, in their mind, is this person with a high focus score and a ocular staining score less than 4 more likely to have Sjogren's than this person? And so they would choose one of the two vignettes, or they would say they're equal. And they respond to maybe 30, 40 of these vignettes, and in the end, you basically get a weight attributed to each of these items, depending on how often an item was chosen by an expert. And so this is a flow chart that sort of describes the entire process. So we were fortunate. We did not have to start from scratch. We had all the items that were included in the ACG criteria. We had all the items included in the ACR criteria. So we put those together, and we got together with our expert groups, our 52 clinician experts, and decided on sort of the final list of items that was going to go into this exercise. And then we used the Southern Mind, the multi-criteria decision analysis survey. And that essentially, when it says item reduction, it essentially dropped some of the variables that were almost never chosen, if you will, in the decision made by the experts, and it attributed a weight to each of the items. So now we had kind of a draft criteria, which was strictly based on a model, right? And then that draft criteria was tested and adapted in real patient data. So we got data from SICA, from Paris Sud, which is Xavier Mariette's cohort, and then from the Oklahoma Medical Research Foundation. So we got, as you'll see, 400 observations from each of them. So we had actually 1,200 data points from real patients. We divided these in two at random, and one data set was called the development data set, and one data set was called the external validation, or the validation data set. And in the patient data set, we use clinical vignettes extracted from each of the participants, and we had expert clinicians essentially attribute a clinical diagnosis based on a clinical vignette. And I'm gonna show. So this is how the case and control. You have to have case and controls when you try to assess performance, and it'll be a bit more clear when I show you the survey process. So this is the result of the multi-criteria decision analysis. You could see that it comes up with this, this sort of spider chart, and it attributes essentially a mean percentage or a median in parentheses. And you could see that the highest weights are for the labial salivary gland with focus score and for the anti-SSA and B. And then we get the next most important items are the OSS, the Schirmer test, and the unstimulated whole saliva. And then oral symptoms carry very little weight. So basically, experts did not think that oral symptoms were particularly important. So then the second exercise, once we had done a multi-criteria decision analysis, which again is just based on expert opinion, based on vignettes, right? This is not on real data. Now we utilized our real data. So we got, as I said before, 400 participants from each of these cohorts. So we had 1,200 participants with complete data. And then we generated clinical vignettes from these patient data, and these were randomly divided across surveys. So each survey included 46 clinical vignettes, and the clinical vignettes, would include things like demographics. I mean, they basically almost like a patient chart. If you get to look at a patient chart with some test results, when you have enough tests in there, you could say, well, this patient looks like they have Sjogren's or they don't have Sjogren's. And each of these surveys were administered to 2 clinician experts. And of course, they were blinded. They did not do this together. And so we had a total of 26 survey. And in the end, so here are our development data set and our validation data set, and they're divided in 2 at random. And then we administer all of these vignettes basically to 2 clinician survey to 2 clinician expert per survey. And so we came up with a number of concordant responses that led to a number of cases and controls. And then we had, some where they were not sure. And then we had a number of discordant responses. And then discordant ones, when the clinician expert responded something different, we brought in a 3rd clinician expert to basically cut the tie. And that brought us an additional number of case and control. And then we had a number of discordant cases here that became our difficult, what we called our difficult cases. And we also rated those, bringing in basically the difficult cases were graded by 8 members of the steering committee. And that gave us also a number of case and controls based on the difficult cases. So now this is actually this statistical approach called random forest classifier, which is applied to all the clinical data. So you could think of it as it's a little bit like the multi-criteria decision analysis, except it's based on real data. And so when putting in all of our tests, you could see that the ones that sort of came out here were OSS, anti-SSA, and focus score. These were the most important, basically based on this random forest classifier. And if we made the data binary, so in other words, we all of a sudden gave thresholds. We now had the unstimulated whole saliva, the Schirmer test, the OSS, anti-SSA, and focus score that were the most important variables that were used, if you will, by the clinician experts when they rated the vignettes. And so we came up with, so I showed you the spider chart. These are all the percentages that are the weights, if you will, that are obtained from this multi-criteria decision analysis. Then using logistic regression model, in addition to that random forest, we also came up with a set of weights based on real data. And you can see that even though it's a different unit, it's proportionally very, very similar to this, actually. So the only thing is that we didn't really want to have half a point here. So we ended up using this adapted. So from these 2 things, we essentially drafted these criteria where the focus score was going to have the focus score and the anti-SSA were going to have a weight of 3, and the other 3 items would each have a weight of 1 for a total score of 9. And we then measured sensitivity and specificity, as well as the kappa values for sort of candidate scores. So we looked at the multi-criteria decision analysis, the logistic, and what we call the adapted. And we found that when we had a cutoff of 4, we basically had fairly good both specificity and sensitivity with a pretty high kappa value. So that basically became our draft criteria to be tested in the external, in the what's called the validation data set. So essentially, we decided to only use anti-SSA based on the Bayer paper, and of course we had some inclusion criteria, and the draft criteria stated that Sjgren disease, or at the time Sjogren syndrome case, would be defined by a score 4 or greater. And 98% of our clinician experts agreed with this draft classification, which we then ended up testing in the validation data set. And as you can see in the validation sample, we got very high specificity and sensitivity. And we also tested it in what we call the difficult cases. And you remember that the difficult cases that we had from the vignettes that required more than three clinician experts. We also had pretty good sensitivity and specificity in there. And this is basically the final criteria, which were published in 2016 in arthritis and rheumatology, and which are now basically the current classification criteria for Sjogren disease. Now, are these ideal? Well, they're based on consensus criteria. So would it be better to have a biomarker for classification? Sure. But in the absence of a current biomarker or a single blood test, or this is basically the best we have, and it's certainly in par with other diseases such as SLE, RA, or systemic sclerosis. So now that we had done classification criteria, well, I do need to give you one little comment about other work that was done as part of SICA, and that was led by my colleague Lindsay Criswell and Kim Taylor, where they examined the genetic etiology of Sjogren disease across ancestry and disease subsets, and were able to do that because, as you recall, DNA was extracted from every single from whole blood from every single participant. So they did a global GWAS and a subpopulation GWAS in 2 of the largest ethnic groups, the European and the Asian. And they found significant association in established regions of the MHC, and several novel suggestive regions were observed. And genetic associations with Sjogren disease differed markedly according to ancestry, which were not explained by differences in subphenotypes. And what this group also did, they actually used DNA methylation, and they did a genome-wide case control study of 26 SICA participants, and they found almost 8,000 differentially methylated positions, or DMPs, that were associated with disease status. And 57 genes that were enriched for DMPs in their respective promoters were identified, and many of these were involved in immune responses. Then another analysis, a genome-wide DNA methylation study of the salivary glands, or labial salivary glands, on 131 SICA participants, identified 2 major clusters of Sjogren disease cases with distinct phenotypic features. And both of these papers have been published, and you are welcome to look them up, because they are another important lesson learned from the SICA registry. So now that we have classification criteria, and we still have a very large number of biospecimens in the registry, or in the biorepository, we thought, well, we could get so much more out of that registry. And we still, because Sjogren is such a heterogeneous disease, both from respect to the phenotypic features and from the standpoint of the inflammatory pathways, there's basically no effective systemic drugs. We need to have a more personalized and targeted approach to treat Sjogren disease patients. We know that gene expression profiling studies are useful to investigate rheumatic diseases with poorly understood idiopathogenesis. And in these transcriptome studies, we have bulk messenger RNA that can explore differences in gene expression between conditions, so Sjogren versus non-Sjogren. And then we have the single cell mRNA sequencing that can help us understand which genes are being expressed at the level of individual cells, so we can better understand the diversity across the tissues. For example, in the PBMCs, across immune cells, and in the labial salivary glands. And so we obtained a new grant. And again, I apologize, the format of my slides shifted a bit here in the transfer. But we obtained a new grant, which we call now SICA Next Generation Studies, or SICA Next Gen. And this grant is going to go from 2020 to 2025. It's a U01. And the goals of the grant is to generate transcriptomic profiles, so bulk RNA-seq, single-cell RNA-seq from our PBMCs, and single nuclei RNA-seq from our labial salivary glands, so we can better understand the diversity across subsets of SICA participants and across cell and tissue types. And so the hypothesis that we'll identify some clusters of participants with similar omics profiles, and that these clusters will be associated with specific Sjogren syndrome phenotypic profiles. We are also interested in exploring the correlation between omics profiles and signs of severe disease manifestation. So as you know, the presence of germinal cell formation, and the FLS was a high focus score in the biopsies, as well as high IgG and low complement. Those are all signs of severe disease manifestations. So the hypothesis is that omics profiles will identify etiologically related clusters of Sjogren disease patients with severe disease manifestations and corresponding biological targets for treatment research. And then we also will have an expanded dissemination plan, because we're going to make these omics data set available to investigators worldwide. And that, since we're in process of generating the omics data set, obviously this is not yet available, but it will be very soon. It will be at some point in the next few years. So in this work, we're collaborating with the UCSF Collabs, and Collabs brings under one umbrella groups like disease to biology, flow cytometry, genomics, and data sciences. And we're mainly working with the data sciences directed by Gabi Fragedakis and genomics with Walter Eckelbar. And these are all our various collaborators within Collabs, within SICA. We still have a dissemination plan, as I mentioned, and then we have an external advisory group. So this is basically sort of our assay workflow. In our biorepository, we have our PBMCs. The PBMCs basically go to Collabs for all the different sequencing work, the single cell RNA-seq, the bulk, and the microRNA-seq, and then all of this data goes into data science Collab. And then our salivary glands go to undergo DNA methylation in the Barcelos lab at UC Berkeley. And then they go to the Knox lab, where this work here is still kind of in its infancy. So right now they've tried to sequence about 10 glands, but they're essentially running into some issues where when you thaw frozen glands, depend on how they've been frozen, you can't. Actually, there's a lot of cell death, so you can recover nuclei, but not cells. So they're still working on refining this, and we're still kind of at the very beginning of this. So this is really just as an illustration. We don't really have extensive results yet, but just to show you that based on, the bulk RNA-seq analysis on the first 80 samples. So since then, we now actually have run, I think, another 2 batches of 96, but this is based on the first 80. You could see that there is a differential expression of genes that show a cluster of Sjogren patients with high ACR ULAR score, and they're sort of here in the darker red. And these people are mostly positive for SSA, SSB, rheumatoid factor, and NANA. So again, these are the preliminary analyses that are ongoing. This is for the single cell RNA sequencing. We're still at the stage of what's called quality control. So there are a number of artifacts. So these UMAPs are a way to essentially reduce, it's like dimension reduction. So it's a way of making the data sort of interpretable, if you will. And there's a number of quality control steps that have to be taken. So this is basically the stage where we are. We're not yet at the stage of great interpretation. We're still at the stage of quality control. And I'm just showing you this as part of the process. So, and I'm almost done. I realize I tried to not speak too fast. So it went a little longer than I expected, but I'm almost done. The Accelerating Medicines Partnership is a brand new public-private partnership between NIH and then a number of pharmaceutical companies. And the idea is to transform the current model for developing new diagnostics and treatments by jointly identifying and validating promising biological targets for therapeutics. So that whole group started that whole effort started in 2014 with Alzheimer's disease, type 2 diabetes, and RA and lupus. And they joined other diseases, or they added other diseases along the way. And then in 2021, there was a new funding opportunity announcement for what they called AMP2, autoimmune and immune-mediated diseases. And that included Sjogren's disease. So we happily applied to this. And we actually were very, very fortunate to obtain the grant. We just heard it very recently. So this is our group. Again, we're going to be collaborating with OMRF, the Oklahoma Medical Research Foundation, Johns Hopkins, NIDCR, UC Berkeley, and NYU. And the idea here is to essentially utilize our historical cohorts. So together with OMRF, we have more than 5,000 participants recruited between the two of us. And we've had a number of specimens on these participants who are generating multi-omic data. And this will be an opportunity to recruit, to bring back basically participants that will now be 10 to 15 years after the initial evaluation. And we're hoping to learn a lot from these recall participants. And then we will also be recruiting new participants across all these sites, including people with overlap between Sjogren's disease and SLE and RA. And we are hoping to include newer assessments, like, for example, the salivary gland ultrasound. And our scientific themes, well, based on essentially next generation sequencing approaches, both on PBMCs and on tissue. We are hoping to better understand the phenotypic and molecular heterogeneity of Sjogren disease, as well as the disease mechanisms that are inherent to progression, both from non-Sjogren's to Sjogren's and from early to advanced Sjogren's, and with the final goal to identify therapeutic targets to stabilize early disease or reverse and improve advanced disease. So SICA, as I'm sure you'll agree with me, is a very unique data registry and biorepository for Sjogren disease. After a 19-year journey, much has been learned from the SICA cohort about the phenotype of Sjogren's, and we continue to learn a lot about the genotype. And with Sika NextGen, the transcriptome and epigenetic profiles will enrich the Sika registry in the biorepository and will make it a much, such a richer data, such a richer resource for not only for us doing the science within UCSF, but also for investigators around the world who can obtain some of these omics data. And then the latest project here, STAMP, will enable us to interrogate the tissue and systems biology of Sjogren's using these deep sequencing and other cutting-edge genomic technologies to better understand the pathogenesis and ultimately identify therapeutic targets, which is really everybody's goal since we still don't have that with Sjogren's. So I will stop here, acknowledging obviously a very large number of people. Lindsay Criswell, my former co-PI, Lindsay and I became PI of Sika in 2010, and former PIs Daniels and Greenspan, of course Stephen Shiboski, who's done all of the analyses that have gone into the classification criteria, and all of our directors here for the Sika research sites. And then, of course, all of the Collabs, and I shared this with you before, all of my collab collaborators. And I'll end here, and I'll be happy to answer any questions you might have.
Thank you, Caroline, for the excellent talk and very informative presentation. I'm going to check for.
And I apologize for maybe going a little over time. I covered a lot of stuff, but it's been a long project, a 19-year journey. So
yeah, you summarize it very well. Thank you. Peço aos colegas que, se tiverem alguma pergunta, que possam postar aqui na seção de comentários. E, well, I have a very long list of questions, and I would like to ask you while our colleagues writing them, theirs. Initially, it's a very frequent situation that I deal with is when to biopsy a seronegative patient. So especially those young women that just present a reduced resting salivary flow rate and exerostomia, sometimes with arthralgia or mild symptoms, but complete no serologic marker was present. So sometimes, what should we do to improve the pretest odds on this case?
So, you know, the latest
before biopsy, before biopsy. So
yes.
When should I biopsy this patient?
Well, so first of all, the biopsy has become much less invasive than it used to be. So we now can get, five glands out of a frankly 4 to 5 millimeter incision. So if you follow the right, the right methodology, it is really not a particularly invasive procedure. In clinic, I have to admit, I will always run the less invasive test first. So let's say somebody is anti-SSA negative, but I measure the whole saliva. If their whole saliva is less than 0.1 ml per minute, I also measure the stimulated whole saliva. And the stimulated whole saliva, I think, tells us a lot, because it would be very unusual for somebody with Sjogren's to be able to produce 10 ml of stimulated whole saliva in 5 minutes. We often see that, I think, with patients who are taking anticholinergic drugs or who have anxiety. They will be very low on the unstimulated, but they can produce abundant saliva in the stimulated whole saliva. And if I get a lot of stimulated whole saliva and I have a negative serology, I might order the eye, the ocular staining score and the Schirmer. And if none of those are positive, I typically will not do the biopsy. Granted that I have high stimulated whole saliva. If the stimulated whole saliva is also very low, then I get very suspicious about Sjogren's, even in the absence of anti-SSA serology. And I may do the eye test first, but I will do the biopsy if I have both very low stimulated and unstimulated whole saliva. Does that answer your question?
Yes. So do you use the salivary gland ultrasound to select these patients, or is not a routine to you?
So we have not. So our colleagues in Europe are now using salivary gland ultrasound very, I think very commonly. We have not yet gotten to that, but we definitely want to make it part of STAMP, of the Sjögren Accelerating Medicines Partnership. So as we start this new cohort, we definitely will have the ultrasound be part of it, because we want. So a number of papers have showed, I think, the good diagnostic performance of the ultrasound, but in a way, to be fair, you have to almost redo all of the classification criteria analysis, including the ultrasound. And STAMP will allow us to do that. Now, STAMP is not about classification criteria. It's really about biological markers, but we will have the data. So if we have the data that includes the ultrasound, we can very well assess what is the weight of the ultrasound in that algorithm.
Great, thank you. Dr. Felipe Fonseca asks us, given the heterogeneity of Sjogren syndrome, do you think we can achieve an acceptable inter-observer concordance level in the diagnosis of Sjogren syndrome following the current recommendations?
So remember, there's a difference between diagnostic criteria and classification criteria. Now, the classification criteria are really used to basically select patients for entry into clinical trials or clinical studies. And for clinical trials, we really want to make sure you don't misclassify them. So I think the classification criteria tend to be a bit more stringent. However, I have to admit, I use them in clinic because I just find it makes it a lot easier. So I think, if you use them for diagnosis, I think they're fairly, they're fairly reliable. But where we I think run into questions is in much older patients. Sometimes in very old patients, they almost lose the FLS with focus score. And it's almost as if their glands become so sclerotic that they get chronic sclerosing sialidinitis. And you don't always know where these patients' prior FLS was a high focus score or not. So I think sometimes age can be an issue, and it might be difficult to do it from age. Or an older patient might actually have a positive FLS. And it may not necessarily mean that they have, that they have Sjogren's. So the criteria are not perfect by no means, but it's the best we have in the context of current classification criteria for rheumatic diseases.
I have the impression that even though the criteria hasn't changed, the histopathologic analysis of the FOX score and the definition of the FOX score, it's still the same.
Yes.
But it helps us to better understand how to define a focus score. So in how to define the focus score, what is a focus score and what is not. So it leads to here in Brazil, the Brazilian Association of Rheumatology also have printed some guidelines to report biopsies. So it's very helpful to the clinician to see not only the focus score, but also some other aspects that maybe probably because of this publication, then it's now a standard.
You're absolutely right that the training and calibration of the histopathologist who's going to read the slides is incredibly important. And unfortunately, I think that varies a lot. If you have general pathologists who read these slides, they don't do it a lot. It's just, there's going to be a lot of missed diagnosis there. So you really want oral pathologists, I think, to read these slides, and oral pathologists who actually are interested in it and have read the different publications, I think, on FLS and focus score. So you're absolutely right. It's very important to have a well-trained oral pathologist read these slides.
We have another question from Dr. Vitor Martins. Is it recommended to carry out the investigation of all criterion even if during the investigation of a primary Sjgren syndrome the patient has already obtained a minimum score of 4?
So that's a very good question. It's a very good question. It comes back a lot because rheumatologists love to be able to do the diagnosis without having to do the biopsy. And so very often, if they get an anti-SSA positive and the patient has, low unstimulated whole saliva, great, you have your diagnosis. For me, as an oral medicine, clinician, I feel that there is a lot of information in the salivary gland biopsy. So if you have presence of germinal centers, if you have a very high focus score, you know that patient is probably at higher risk for lymphoma. So personally, I think that even though, you don't have to, the patient meets the criteria, I think from a patient management standpoint, if you have a patient with Sjogren's, shown by an anti-SSA and low unstimulated whole saliva, I think it's very important to get their focus score at baseline, and maybe even 5 years down the road to make sure that they're not developing germinal centers or their focus score is not going up, and so I think it's important.
Thank you. And we have another question from Dr. Felipe Fonseca. Do you think IgG4 related disease should always be considering the differential diagnosis of Sjgren syndrome?
Well, so actually, and now I believe it's actually one of our exclusion criteria. So yes, it should be considered, and that's also another reason why the biopsy is important, because the histopathologist can look for features of IgG4 disease on the labial salivary gland biopsy. So yes, I think it should be ruled out.
So, but in this sense, should we also request IgG4 levels and during the investigation of Sjogren's? Do you think it's adequate?
Well, so I think that's often done, on the rheumatology side, because it's and I don't. I think actually IgG4 disease is one of our exclusion criteria. I all of a sudden, I'm sort of. Let me see. Is it? Oh, I don't have the exclusion. But we have a whole list of exclusion criteria, and I can't remember actually. I have to go back and look.
Yeah, I'm quite sure that it's listed there.
Yeah, I think so. So if it's listed as an exclusion criteria, we obviously have to. It means we have to look for it.
Okay. We have another. Can we go just for a few more minutes? We have some questions yet. So, Dr. Tatiana Kimura, she's asking about very old patients, how much the sensitivity of the biopsy should be affected. Dr. Tatiana is a neuropathologist, and she also one of the authors of the Brazilian recommendations of the Brazilian Rheumatology Society recommendations for diagnosing Sjogren syndrome as well.
So that's a very good question. And as I mentioned before, I think that there is a concern that in very old patients, things may look a little different. So I think as we collect, more data, for example, as part of these newer project, that may be something we're able to refine. At this point, we have not really considered age in our classification criteria. We basically have run through the methodology, but have not considered age. But I think classification criteria, are constant. They constantly undergo improvement, right? And so I think in the next iteration, when we start exploring the salivary gland ultrasound, I think it would be really a good idea to take, basically consider age and do a sub-analysis of patients, maybe, 85 and older. Is this a different patient population? So at this point, I can't really answer your question because we haven't looked at age, but I think we should be doing that by doing a sub-analysis in older patients. I think that would be a very good idea.
But is it also, regardless of age, if the disease duration, does it affect it may affect it similar than age? Like a 25-year-old patient who starts Sjgren's when she's 55, maybe she can have similar alteration than an 85 patient with Sjgren's who started at 70 or 65.
That's possible. But as you know, it's very difficult to first of all, until we had good classification criteria, people were often diagnosed much later. And so the essentially the duration of disease is often really hard to know, because they might start with having, mild to moderate dry mouth, and that could actually take years until they get tested. So I think as criteria get used more commonly, we'll get a much better picture of duration of disease. I think at this point, it's still, we don't have very good data on duration of disease. Like in Sika, we asked them, we asked participants in our questionnaire, when did your symptoms of dry eyes and dry mouth start? And you get kind of an estimate of, 5 years ago, 10 years ago. It's the longer it is, the harder it is for patients to remember. So I think now that we're applying the criteria on many more people, we're going to start getting better data on duration of disease. But it's not something that we have so far.
Okay, we have two more questions, then okay, three more questions, then we're finished. Dr. Fabio Pires asked us, do you think previous steroid therapy would influence histological analysis?
So actually,
or even biologics.
Yeah, that's a very good question. And I did not show you the detail of, if you actually read the paper, the follow-up paper that we published, we actually stratified patients by whether or not they were receiving any immunomodulating drugs, and that actually included steroids. I mean, we kind of lumped all of the immunosuppressants and immunomodulating drugs together. And we did the follow-up analysis comparing patients baseline to follow-up in the different categories, whether they were taking these drugs or not, and whether they were taking them at baseline and at follow-up, or neither at baseline or neither at follow-up, or only at baseline, and actually found no difference. Found no difference when we looked at focus score. There were about, I would say in each of these categories, there were about 7% to 9% of participants who had FLS was a focus score at baseline and no longer had it at follow-up. But we don't really believe, and this was independent, regardless of whether they were taking treatment or not. So we observed that in people not taking any treatment at baseline or follow-up. So we think that 7 to 9%, that's basically sampling error. So in other words, if you collect salivary glands, well, you might at time collect glands where you can't see the you can't see the FLS. So I think there is some sampling error going on. We noticed a huge difference in size in the salivary glands that were collected. So as you know, you need 4 square millimeter of gland tissue to basically assess for FLS and focus score. And in very small glands, it's harder. So then you have to kind of put all these small glands together. So I believe that the 7 to 9% who regressed are very likely to be sampling error. But we don't know. But it was not associated with drugs. That can tell you that.
Yeah, if during the last workshop, we had the opportunity to work with systematic review on biologics on Sjgrens, and we couldn't find this information. But some papers evaluating biologics, some of them, very few actually, reported that especially rituximab may change, but it's not a consensus yet. So we don't know. We need to observe it more.
Yeah. So in Zika, remember Zika started in 2003. So I don't think a lot of people were on biologics then. So a lot of the drugs that were taken then were probably more like hydroxychloroquine, prednisone. There was some biologics, but it wasn't as common as it is now. And also biologics have not shown, like rituximab has not shown to be effective for Sjogren. So I don't think a lot of rheumatologists prescribe rituximab, frankly, to manage Sjogren's, unless they have another autoimmune disease. They have another rheumatic autoimmune disease.
So we go back to Dr. Wagner Gomez's question. Congratulations, doctor. Do you have any experience in Sjogren's like adverse events in patients undergoing immunotherapies for cancer treatment?
That's a very good question. And this is kind of anecdotal. I've seen a number of lichenoid changes, very drastic lichenoid changes in the mouth in patients undergoing immune therapy for cancer. I haven't really seen Sjogren, but that's just my clinical practice. I mean, I can't say we haven't studied this. It's not like we have a cohort studying that. But I think it's a good question, because if you can see lichenoid changes, I'm sure some of these patients might also have Sjogren's like adverse reaction. And interestingly, the 2 patients I can think of who had very, very their whole mouth was covered in lichenoid, it looked like very severe, like implanus. It actually faded away after about, I would say, 10 months following the discontinuation of the immune therapy. I had to treat them pretty aggressively during the immune therapy and for 10 months after the end of the immune therapy. So it eventually faded. But I can't really answer your question about the Sjogren.
If I'm not wrong, Blake Warner from NCDR, who seems to be your collaborate with your new project, he has a very interesting publication and maybe a cohort of patients with Sjogren's like symptoms in patients with undergoing immunotherapies. So do you plan to at some point include these patients on the analysis, or if they really have Sjogren's following immunotherapy, or if it's just like a symptom? Because it has been described that some patients may have even positive biopsies and positive serology.
So that's a good thought, but I think it'll depend whether or not we think that group of patients can help us identify good therapeutic targets and better understand the pathogenesis of the disease. So at this point, probably because it's probably relatively rare, I would imagine we would exclude such patients from, from the stamp cohort, but we have a whole year to sort of define our scientific agenda. So I think as we start discussing our scientific agenda, we definitely, I know that there's a lot of talk about COVID, how are we going to treat COVID and how are we going to take COVID vaccination, COVID infection into account in recruiting these participants, because it's become so common now and it's endemic. So we feel that we have to basically measure the history of COVID infection and whether or not they had vaccination. And so I think anything common like COVID will be taken into account. And also because COVID does cause taste alteration, sometimes dry mouth. I mean, so there are a number of potential questions there, but I don't know if we'll have the opportunity to also include, patients with immune therapy for cancer, because they, typically they would have other issues that might confound the main study question. So I don't know yet is the answer.
We have a last comment from Dr. Alan Rogers Santos Silva. Thank you, Professor Shiboski. Any chance you could mention your vision about the impact of the World Workshop on the future generations of oral medicine?
That's a good question, because we're right in the middle of trying to do a survey. I think the fact that Luis and I were invited to basically oversee this working group with that very question: what are the barriers that prevent oral medicine research? And so the fact that we were asked to do that, I think that's a very good sign, because it means that there is an awareness and an interest in measuring. Well, so what are the issues with colleagues, oral medicine colleagues doing research? And I mean, if I had to guess, I think the short answer is education. If you just have clinical trainings, clinical residencies that do not include, like a PhD or a master's, you're not going to have research by people who train in clinical residencies. And so I think a lot of it has to do with the ability of institutions to get I've spent 30 years in the US, so my answer is going to be very US centric, but if you can get training grants from the NIH, like K-12s and K-type grants, to basically encourage your residents to do a PhD, I think that's what will develop more research in the field of oral medicine. I think a lot of it has to do with the original training.
Yeah.
And I'm interested, Luis, if what is actually your feedback on that? I mean, you can answer this question as well as I can.
Yeah, I agree with you regarding the need of training. We have a very interesting situation regarding training here in Brazil, because even though we don't have oral medicine residencies, our clinical training is made during the master's and the PhD. So it has a strong scientific background. So most of the trainees are exposed to research, and the large majority of them still want to do research, even if they are not affiliated with an academic institution. So even they go to hospitals, or even if they are in a private practice, they tend to be in contact with research at some point. So it probably because of the background and the formation.
And actually a lot of literature in oral medicine research comes from Brazil. So my other hat is in HIV. I do a lot of HIV research, and I have over the course of my career. And there is a very high proportion of the literature on HIV, oral HIV, that comes from Brazil. And so I think it's going to be very interesting so right now we're doing this survey among oral medicine experts around the world to sort of see how much research is being done, you know, and what are the barriers. And I think when we analyze the data by country, I would bet that you're going to find a lot more research being done in Brazil than in other countries. And other countries might, I mean, I think, you know, we'll see some pockets coming up of you know, research being done. But I bet you in the US, there's actually not going to be as much, because in the US, the majority of our oral medicine residencies are clinical residencies. And I can see it trying to recruit oral medicine faculty is actually really difficult, because most people applying to these positions have no background in research whatsoever. So I bet you we're going to see a lot more research training and research, you know, experience in non-US countries. And I think Brazil is one that probably will have the highest, I would guess.
One of the aspects, just commenting this question, that the World Workshop specifically has evaluated some other aspects than research. They have previously evaluated, like the training neuromedicine, like in previous edition. So and now they have asked another questions before, and how they training, the global training neuromedicine, and now they are asking what the barriers to research. So in this sense, we try to, the group tried to lead the oral medicine community in a way that we can improve the standards of now initially the training, what should be an ideal standard of training, and now what's the standard for research in a global sense. So it's very
it's a great step forward. And I think the WWM is now, you know, much more focused on research than maybe it was, you know, or I shouldn't say more focused on research, but I think there is a lot more incentive to try to actually see what can we do to change the situation. I think there is a general recognition that you know, there's only a very small number of people who are doing research in oral medicine, and how can we change that? So well, thank you very much for inspiring questions.
Thank you very much. It was a very lovely afternoon, and thank you for the opportunity to share your view and your experience with us. And thank you to the Brazilian Society for these meetings every once a month on Saturdays. It's always a good opportunity to learn and to hear from people around the world. Thank you all. We're going to finish this, and the talk should be available on the YouTube of our society. And thank you all.
My pleasure. Bye-bye.